All right. Well, it is 1 o'clock in the Central Time Zone, so we will get started. Thank you so much, everyone, for joining us today, and welcome to the Monarch Conservation Webinar Series. Today's webinar is Milkweed and Floral Resource Availability for Monarchs in the U.S., 8 years of Integrated Monarch Monitoring Program data, presented by Jennifer Thiem and Sarah Gomes from the Monarch Joint Venture. My name is Jenna Tots. I'm the Education Coordinator at the Monarch Joint Venture. We want to thank our sponsors real quick for their support of this webinar series, supporting sponsor Reinvent Your Waste and community sponsors East Central Energy, Hedgerow Farms, and NiSource. We appreciate your support so much and your dedication to monarch conservation. Before we get started, there we go. Before we get started, here are a few community guidelines and helpful hints. During the webinar, please put your questions in the Q&A box found at the bottom of your screen. We will collect those for the Q&A session at the end. You are able to use the chat, but it just chats to the speakers. So please put questions in the Q&A box. Please use your reactions, also found at the bottom of your Zoom screen. The speakers can see them, and it is so fun to celebrate what they are talking about. This webinar will be recorded. It is being recorded as we speak and be posted on the Monarch Joint website by Friday of this week. The entire Monarch Conservation Webinar archive is free and available to watch. It has over 9 years of really amazing monarch conservation content, so please check that out. If you're joining us today but did not register for the webinar, we would like to count you for attendance. Please send me an email at learn@monarchjointventure.org with your name and email address, and we will get you counted. Thank you. We want to remind you that we still have the translated captions for Zoom. If you would like to add captions in a language other than English, click the arrow on captions and be sure the translation button is on, and then you can choose your caption language. Not forgetting that you can absolutely add English captions as well. And finally, all of the webinar speakers represent their own viewpoints, which are not necessarily those of the Monarch Joint Venture. However, this week, I believe we will share the views of our fabulous speakers. And I would like to now introduce those speakers. First, we have Jennifer Thiem. She cultivates the MJV's network of researchers, research entities, and partner science initiatives to collaboratively advance monarch conservation research. She also coordinates MJV's research and monitoring projects and works with MJV partners to increase engagement in and sharing of monarch research. Jennifer works with the MJV science team to maintain the Integrated Monarch Monitoring Program, encourage its use, and share findings from the program from the broader conservation community. Jennifer received her BS in Environmental Science from Carroll University in Wisconsin and her MS in Fisheries and Wildlife Biology from the Ohio State University. Sarah Gomes supports MJV's scientific research initiatives to advance monarch conservation in the West. Sarah coordinates MJV's on-the-ground monitoring work. Sorry, Sarah, we need to switch to the page. Sarah coordinates MJV's on-the-ground monitoring work in California, surveying public and private lands throughout the state using the Integrated Monarch Monitoring Program. Sarah provides various services for MJV's science projects and supports MJV's strong network of partnerships. Sarah earned a BS in Environmental Science and MA in Geography from San Francisco State University with a focus in historical biogeography, plant pollinator interactions, and plant ecophysiology. I will stop sharing my screen and hand it over to our fabulous speakers. Thank you so much.
All right. Thank you, Jenna. Thanks for your patience while I pull up this screen here. So I'm excited to be here with you and to be here with Sarah, who is also working with the Integrated Monarch Monitoring Program dataset. And so here today, we have the opportunity to share some findings with you from this data set. The program's been going for over 8 years now, and we finally had some research published looking at that whole data set, as well as working on some smaller data sets as well from it. So we'll take a step back real quick and just point out that we're here today and this program is here today because of the monarch butterfly, which is present throughout the world and has both migratory and non-migratory populations. And a large part of why we're here in this place today is because the monarch populations overwintering in North America have been dramatically declining in recent decades. So they've been dipping to near or below certain population thresholds. And in order to stop or reverse those trends, there are goals in place to add habitat to both the eastern and western breeding areas. So on the left-hand panel here, you see a small bar graph in orange, and these bars are graphing the overwintering population of the eastern North American monarch population, so which that resides and migrates primarily east of the Rocky Mountains. And that population has been declining over several decades, reaching its second lowest number this past winter. On the right-hand side, there is a graph in green with green bars showing the counts of monarch individuals observed overwintering in California on the West Coast. And those two have been declining, particularly when you compare them to the blue line that is going upward through this graph. And that means that more sites have been surveyed for Western monarchs overwintering, and yet fewer individuals are being found. So in order to combat those declines in the east, there's a goal to add 1.3 billion additional milkweed stems to the landscape. And in the west, there's a goal to add 50,000 acres of habitat, which would include both milkweed and nectar resources. That said, there's still substantial data needs in order to better understand why monarchs are declining. And then with these goals in place, there's a need to be able to track our progress in putting that habitat on the landscape, understand where there's potential to support more monarchs, and a lot of other research needs and data gaps. And so to answer some of those questions, or at least define a method of how to gather the data that can answer these questions, a group of scientists and land managers and conservation organizations from across North America started gathering in 2015. For several years, they met frequently and identified those data gaps and methods of gathering data that could answer questions like, where is the milkweed growing? Where is milkweed not growing? What land use types like roadsides or developed spaces or protected grasslands are the monarchs using? And where are the best places to target our efforts? So out of that was born the Integrated Monarch Monitoring Program. It's a national initiative designed to monitor both monarchs and their habitats in order to evaluate conservation efforts and compare habitat and habitat use by monarchs in different land use types. One key to this program is the focus on random sites. So there are several other data sets, large data sets on monarchs that tend to be from self-selected sites and may not be representative of what's on the landscape. So a key to this program is to collect data on randomly located sites. And so this Monarch Conservation Science Partnership, shown previously, generated a map of spatially balanced, randomly selected sites where habitat may potentially exist, like randomly selected roadsides, random agricultural borders, random open spaces. And data from those random sites contributes to that unbiased picture of what's actually going on across the landscape. However, we can still gather important information from non-random sites. For example, a person might want to track how monarch use changes on their site as they manage habitat. So in those types of cases, the IMMP, the Integrated Monarch Monitoring Program, can also be conducted on non-random sites, and that provides some really important information too. And then data from all those types, the random sites and the non-random sites, can be combined when appropriate to answer certain research questions. And then we have this holistic, expansive data set. Just a very brief picture of what that IMMP entails. So there are several optional activities when performing the IMMP. This is something that community scientists can do, biologists, academics working on particular research projects. So one component of the IMMP is a search for milkweed and blooming plants. And there's a specific way and a specific space that those are recorded. so that the data are comparable among all these sites. There's also an option to look for monarch egg and larvae activity. So looking at milkweed on your site and recording when you find eggs and larvae. There's also an option to record adult monarch activity. So there's a specific method of where to walk and how fast to walk and what to record with regard to monarchs and their activity, adult monarchs and their activity. And then there's even a survival and parasitism option. So there's an option to collect a 4th or 5th instar from your site reared indoors and report the survival and parasitism outcomes. So this is really novel because it combines the milkweed resources, the floral resources, and the monarch activity all in one. So although there have been a lot of options to monitor pollinators at large scales, this is really the most comprehensive option for monarchs and their habitat. And it's also applicable to other pollinators because you're getting that great information on floral resources in a very detailed and consistent way. So this was started to be tested in 2016. It was refined a little bit in 2017. And since then, the over 1,200 sites have been surveyed. We've got a map on the bottom right that shows really spanning from east to west, highlighting a lot of surveys in the upper Midwest, primarily because that's a big core of monarch summer breeding grounds. But we've also got a lot of samples through that central flyway down into Texas and all over the West Coast through California and up throughout the Southwest US as well. It's covered 26 states so far with well over 300 participants. The extent and the distribution of these sites gives us an excellent opportunity to explore monarch habitat in specific places or looking at that whole data set across the US. And so to take a look at this regionally, I'll hand this over to Sarah so she can explain how she's looking at using the IMMP to explore a large Western landscape, which also includes randomly selected points.
Wonderful. Thanks, Jen. Just going to swap screens here. All right. So yeah, like Jen said, we're going to take a moment to just explore how IMMP is being used in the West and give some examples of how data is being collected and then how we're using that data here, particularly in California. So Monarch Joint Venture is engaged in several projects to monitor monarch habitat in the Western US. And a few on-the-ground statewide projects that we have work with the Bureau of Land Management, California Association of Resource Conservation Districts. We're doing IMMP on private working lands, so working with the National Fish and Wildlife Foundation for that, and then a whole number of other partner programs that we support as well. So IMMP is being used frequently in the West, and we're going to use some of our Bureau of Land Management projects as an example. So recently we've had two main projects working with the BLM. The first occurred between 2021 and 2022. And then our current project is in progress between 2023 and 2025. So in that first project, the goal here was really to find and document monarch and milkweed occurrences on BLM lands throughout California. So we used random and non-random sites with a focus on random sites for an unbiased approach to really survey the entire landscape. And then we use that information in our current project to target known occurrences. And here we're establishing long-term monitoring plots and collecting 3 years of baseline data. And our main focus for this presentation is on in these 2 years, our crews conducted 428 unique surveys. That includes the milkweed and blooming plant surveys that Jen mentioned, as well as egg and larval surveys. And then there's a whole lot of information that we collected, and we decided to break it down in chunks. So We analyzed data by ecoregion, by site selection type, by suitability ranking. We examined milkweed and floral associations, so looking at species richness or flowering plant diversity and the frequency of blooming plants. And we also put together lists of common floral species that co-occurred with milkweed. And then lastly, we looked at monarch observations, thinking critically about which milkweed species occurred with monarchs, and then also painting a bigger picture with other environmental variables such as distance to water. All right, so first thing we did was we broke things up by species. So here we have a graph of milkweed density by species. We found that Asclepias fascicularis, or narrowleaf milkweed, grew in the highest density. It was also the most widespread and common species that we found. It's not surprising because it has the largest range out of all the species in California. And that was followed by Ariocarpa. But of course, these species all occur in different regions throughout the state. So we wanted to really look at things spatially. So we broke things up by ecoregion. And there's a lot going on in this slide, so I'm going to walk us through it here. On the left, we have a table summarizing milkweed resources, floral resources, and then further native floral resources by ecoregion. And this is the level 3 classification from the United States Geological Survey. On the right, we have a reference of California with those ecoregions in case you want to compare the two. So first, looking purely at the number of sites, we see that the Central California foothills and coastal mountains had the greatest number of sites with milkweed present. However, we had different number of sites in each ecoregion category. So when we're looking at the percentage of milkweed present within those surveyed plots, we see that the Cascades had the highest proportion of milkweed. The Northern Basin in purple here had the highest milkweed density. And then down below, we see that the Southern California mountains had greatest species richness. And then the Coast Range and Eastern Cascades tied for the greatest floral frequency. And this 0.6 means that about 60% of subplots sampled contained a blooming plant. And then when looking at native resources in particular, again, we see that the Southern California mountains had the greatest floral diversity, as well as the greatest floral frequency with about 90%. Summarize what exactly we're seeing on that landscape. Next, we broke things up by site selection type, so random versus non-random or self-selected, and then looking at habitat suitability within those categories. So on the left here, we've got a table of all plots, random plots, and self-selected plots by suitability ranking, low, moderate, and high. And then on the right, we have the spatial map of habitat suitability and milkweed presence, and the black dots represent sites where milkweed was found present. This represents the DILT's Western Habitat Suitability Model, in particular the non-tropical breeding habitat model. Now moving back to this table, the main takeaway message is that we found milkweed was more likely to occur in higher suitability sites. So the proportion of plots with milkweed present was greater and increased with suitability. And this all suggests that this is a helpful tool when determining where we might want to establish these plots. Moving on to milkweed and floral associations. Here we've got a few graphs. In the top two, we've got blue and green representing species richness or diversity. So on the left, all floral species. On the right, native floral species. On the bottom, in pink and orange, we've got floral frequency, again, the frequency of blooming plants, and native floral frequency. 0 represents where milkweed is absent. 1 represents where milkweed is present. And what we see in all 4 of these graphs is that both species richness and floral frequency are all right. I won't spend too long on this, but I wanted to show as an example some of the lists we created of co-occurring nectar plants by region. So here's an example of some of the lists we generated. On the left, we have all flowering plants, and on the right, we have flowering plants that occurred in sites with milkweed present. And again, we broke things up by region. All right. Next up, we looked at monarch observations with respect to milkweed and also other variables. So what we see here is that one thing I should say is that we had very few monarch observations in these 2 years. It doesn't necessarily mean that monarchs aren't there, just means that we didn't capture them. The timing of our visit and their visit didn't quite line up. We saw that monarchs were observed on 6% of plots containing milkweed, and of those plots, they were observed on red, purple, and green, respectively. One thing that we also noticed by thinking more critically about these observations is that monarchs were often observed in areas with water features nearby. So all of these observations occurred within 340 meters of a water source. And that ranged anywhere from 25 meters away to 340 meters away. But most observations occurred within 140 meters or less. So it's really interesting, and it kind of brings home that point that water is so important here in the West, and that might be driving some of the patterns that we see. All right, and bringing this all home, I'm going to transition to our current research project here. And again, this project, the goal is really to target known milkweed occurrences or monarch occurrences. So we use the 2021 and 22 data set collected by MJV field technicians. We worked with BLM biologists, botanists, and other natural resource managers to identify priority spots or areas of interest, areas with known monarch observations. And then we also used community science data, such as the Western Monarch Milkweed Mapper and iNaturalist, to figure out where exactly monarchs might be on BLM lands. And then we used all of that information to establish these long-term plots. So right now we're in the midst of collecting 3 years of baseline data. And our goal is to represent all 14 field offices from the BLM, all ecoregions, and get a good representation of the state to identify where monarch habitat occurs, and then better understand how, when, and where monarchs use these BLM lands. And with that, I'm going to transfer back over to Jen for some more examples.
Thank you, Sarah. Yeah, that was a great example of using the IMMP for specific research questions, very targeted application on the ground of where they're picking out sites, and she's continuing to grow that data set. I'm going to take a step back a little bit, and we'll talk about broader exploration of the data set. So the results I'll discuss here are from a recently published paper by Laura Lukens, who was a former MJV employee, who's currently a graduate student at Colorado State University, and Wayne Thogmartin, who's a research biologist at the U.S. Geological Survey, and myself. So we used the whole floral and milkweed data set from the IMMP from 2016 through 2022 to explore a couple different factors here. So we wanted to look at those milkweed and floral resources, and then really explore what's driving or what's connected to the frequency or distribution of those resources. So is it based on the geography or different land use types? We did take a look at the random versus non-random effects, and explored a little bit of intra-annual seasonality there. So we took that large data set, which had different numbers of surveys for milkweed versus the floral resources, because the IMMP is so modular that there are options of what people can do when they're on a site. And so some people, most people will survey both milkweed and floral resources, but some will just do one or the other. And so our milkweed data set had over 2,700 surveys for milkweed. Some of them were repeat visits. 37% of those surveys were on random sites, and 63% were on non-random sites. Those surveys occurred across 1,227 unique sites, and those sites were visited between 1 or 21 times per year. The average was 2 visits a year. Milkweed was present at 57% of the surveys that occurred, and 32 species of milkweed were documented. A couple things we can point out here. We looked at this regionally. So actually I'll go back here and just describe this map here is split into 3 kind of color-coded sections here. This is a map generated by the U.S. Fish and Wildlife Service that defines monarch butterfly conservation units. So basically, roughly west of the Rocky Mountains in the U.S. is the Western MCU, Monarch Conservation Unit. The north central and eastern kind of Midwest area of the U.S. is considered the North Conservation Unit. And then south of there, from like Texas to Florida and the East Coast, is the Southern Monarch Conservation Unit. And those are subdivided within their units a little bit, but we looked at those broad categories of West, North, and South. So in terms of milkweed findings, within the Western Monarch Conservation Unit, we found that Asclepias speciosa, showy milkweed, had the highest average density on random sites. So on those randomly selected points, you were most likely to have the highest density of showy milkweed, and then Asclepias subverticillata was the highest density on non-random sites. In the south, the highest density was Asclepias syriaca, and that was on random sites. And then if you went to a non-random site, the highest density was Asclepias linearis. Up in the north, Asclepias syriaca was also the highest average densities on random sites, just like in the south. And Asclepias verticillata had the highest density on non-random sites. So what was explaining milkweed density? We did some modeling and threw in a lot of variables to see what best explained how milkweed density varied across this landscape. And the top model explained 79% of the variation in density. So a model might explain from 0 to 100%, and being able to explain 79% of the variation is very good. So it was a very good fit based on the parameters that we entered in the model. A key parameter was plot identity. So that's basically the plot itself had some heterogeneity that was explained by just the location of where the plot was. We also found a difference in density based on random versus non-random site type. So the random sites had lower milkweed stem densities. The non-random sites had higher milkweed stem densities. That kind of plays into what we suspected. Those non-random sites are often conservation sites. It's where people are choosing to monitor. It's either often where they're putting in milkweed or where they've seen milkweed and they want to monitor to find monarchs. We saw some differences in those regions. So milkweed density was greater in the north region compared to the west. The south, we couldn't really say because you can see that long line in the middle. It was kind of imprecisely estimated. So we had really wide variability of milkweed density in the south. But we were finding higher milkweed stem density in the north compared to the west. We also looked at the site type or land use type. So that was a driver in explaining milkweed stem density. There was lower milkweed stem density on agricultural conservation lands. That's the ACL, the bar on the far left there. Lower milkweed density there when compared to the rights of way, which is up higher, the purple on the right, and lower on ACL when compared to the unclassified grasslands. Unclassified grasslands tend to be private grasslands or grasslands that are not in some sort of protected or conservation status. And then we did evaluate within the year when we were seeing peaks in milkweed densities and saw that across all regions and site types or those land uses, the milkweed density peaked during mid-July. So that was around the day of the year 200. And that's consistent with some other papers that have been exploring milkweed density trends within the year. Taking a look at our floral resource data set that had 2,742 surveys for blooming plants. 41% of those surveys occurred on random sites and 59 were on non-random sites. That was just over 1,200 unique sites where this occurred. And those sites averaged about 2 visits per year, with that range being 1 visit or all the way up to 12 visits per year. The blooming plants, so a plant in flower at the time of survey, that was present at 91% of the surveys. And we documented, or all IMMP participants documented 2,200 blooming plant species. So we were able to explore the kind of lists of frequency. So what species were occurring most frequently in each region. So I've listed the top 3 here by region just for native plants. So in the north, we were most likely to find Rudbeckia hirta, the black-eyed Susan. That second one there is a yellow wood sorrel, or excuse me, a common yarrow, is it a Chile milleforium, and then Monarchs, so bee balm was the third most frequently observed blooming plant in the north. In the south, we had a yellow wood sorrel, black-eyed Susan again coming up at the top, and then firewheel. And in the west, we had a different list. It was a little shrubbier. The most frequently a plant in flower was a creosote bush, followed by the burrow bush and eastern Mojave buckwheat. We also explored this by non-native species. So we've got different lists here, but the most frequently observed non-native blooms in the north were red clover, Canada thistle, and white clover. In the south, we most often found black medic, scarlet pimpernel, and annual ragweed. And then in the west, there was red stem stork's bill, yellow star thistle, and prickly lettuce. And similar to the milkweed modeling, we explored different models to explain the floral resource frequency. The top model here explained only 31% of the variation, which means that our model wasn't great. There is something that we weren't able to account for outside of the factors we collected, like land use and site type and things. But we did find lower floral frequency on random sites and higher floral frequency on those non-random sites. And we found that floral frequency was more than 25% lower in the south and the west when compared to the north. So the north did have a higher floral frequency. And the rights of way had lower floral frequency when compared to those agricultural conservation lands, ACL, or the developed open spaces, DEV, and also when compared to the UGS, the unclassified grasslands. And I do just want to point out that the parameter estimates for the site types from these best models were negatively correlated. So that means that where we were finding higher milkweed stem density, we were finding lower floral frequency. A couple key takeaways here are that the non-random sites were not representative of the landscape. So we were definitely finding significant differences between the random versus non-randomly selected sites. So that's definitely something to keep in mind when exploring this data set as a whole. There's also untapped potential for rights-of-way habitat, kind of a just add flowers scenario here. So where you're finding on roadsides, they tended to have a lot of milkweed, and that's across all three geographies we looked at. And so by enhancing the roadsides, maybe adding a certain management type, integrated vegetation management, a little bit of seeding to promote desirable and native plant communities, you can really boost the potential of that land use type. What interesting piece was that when milkweed was present, the densities were similar among all three geographies. And so although you were more likely to go to a random site in the north and actually find milkweed, if you were at any site in the north or south or west that had milkweed, that milkweed was occurring at about the same density. So that might have some management implications of how you want to maybe spread out or put effort into getting milkweed on the ground. We also found more milkweed on developed open spaces in the north than previously thought. There's been a little bit of research as to how much milkweed is in developed or city landscapes, and this is kind of showing how well they pop in terms of milkweed densities. So there's a lot of opportunity here. This was just scratching the surface of this massive data set. We only looked at floral resources and milkweed density. And as you recall, there's other pieces of the IMMP. So there are management history. So any site that has management applied to it, that gets recorded as part of the IMMP. So that could be a piece that's incorporated, and maybe that would help explain the floral resource frequency, where we weren't explaining it very well with the parameters in our model. You might also consider soil type, annual weather, or distance to water. Like Sarah was talking about, finding that distance to water was actually an important factor for finding milkweed. And that was not a piece of this particular research publication, but could be in future data sets. We can also incorporate monarch data, consider maybe oviposition or larval growth or nectar preferences. All of that is a whole bunch of wonderful information you can get from other IMMP activities that weren't represented in this paper I just talked about. We could also explore seasonality, so variation within or among years. So that's something we can capture. Some of these sites, including some that Sarah's team went out to, were only surveyed one time in a given year. And so there is something to consider about getting back out to sites multiple times to see how those resources change throughout the year and comparing that among different geographies. So I'll just invite you to check out the IMMP webpage as well. It's under the science program on the MJV website. And this lists all kinds of papers that use the IMMP data or its protocols. So these are not the only two research projects out there. This is a data set that is available to researchers. We provide it. Certain data are masked according to landowner preferences. So you can't pinpoint locations of certain things. And this here is a list and a growing list of research that uses that data set. So that data set can be requested via an online request form for formal research projects. We want to make sure that we get those publications up on our website so that we can share that powerful information. And then I just want to point out another awesome resource guide is that IMMP is not the only source of monarch habitat or monarch activity data. And so the Monarch Joint Venture has a data and information working group who pulled together last year a monarch database resource guide. And so this is also within our science program webpage. And it's a list of all kinds of other data sets that have data accessible to researchers that you may be able to pull from if you are exploring questions related to monarch populations and their habitat. So thank you very, very much for joining us today. Glad to share all this information with you, and I'll turn it back over to Jenna.
Great. Thank you so much, Jen and Sarah, for sharing your expertise with us. We'll take some time for Q&A right now, and just know that if we don't get to all of the questions, I will send those over to Sarah and Jen and get those out into an email, hopefully by Friday or early next week. And also a reminder that, again, this webinar will be posted by the end of the week as well. I didn't remind Jen and Sarah that I was going to ask them this question. So if we need to come back to it, I am totally okay with that. But I do need to know what your favorite milkweed is and maybe what you like about it. So if you want to come back, that's totally fine. But if you feel you're ready, we can start there.
Well, I don't feel like a good judge, I'll say that, but I'm very partial to the scent of swamp milkweed.
An excellent choice. Sarah, are you prepared and ready?
I am. Yeah, I was going to say California milkweed is probably my favorite, although they're all fantastic. And the reason I say that is because I love the rich purple color of those flowers, and the fuzziness of the leaves I think are so fun. But that's a great question. Hard to answer.
Excellent. I know. And I have not prepared all of our speakers for that question. I like to ask it, but I need to be better about preparing so people can get okay with their answer. All right, I've got one for Jen here. This was asked about the end of your first section. I'm referring to the IMMP map. And they just asked, does this include MLMP?
No, that map does not include MLMP.
And none of the content that you're talking about today does include. This is strictly IMMP data.
That's right. And that's largely because the MLMP doesn't have like the habitat information in the same way that IMMP collects it. So the IMMP, when you're looking for monarch eggs and larvae, that's actually structured to align with MLMP. And maybe to explain here, MLMP is the Monarch Larvae Monitoring Project. It's a sort of, it's a egg and larvae survey for monarchs that's oftentimes used in gardens and small prairies. And it's been going on, goodness, for 20 some years, I think, based out of jointly hosted between MJV and the University of Wisconsin Arboretum. And it's been around so long, and it's a very successful program that we didn't want to make anything redundant to that. So the IMMP egg and larvae survey is basically the same thing as MLMP, just on a larger scale. So those egg and larvae data could be combined. So anyone thinking about looking at an even larger data set, you could take those two and overlay them. But the IMMP floral data set is much more extensive and doesn't align with MLMPs.
I'll just ask this quick follow-up question. Someone was asking, is MLMP data being analyzed? And if not, why are people still doing it? They are absolutely doing it. And yes, and I don't know if you can speak to that analysis too.
It absolutely is. I couldn't like tell you the most recent pubs off the top of my head, but it is continually used and referenced in research about larval production and trends in monarch breeding and things like that.
Great, thank you.
And
go
Sarah, did you
ahead.
want to jump in about that?
Yeah, I'll add quickly that it's also funnels into the International Monarch BioBlitz as well. That's another recent use for that.
Right. And if you've got, more specific questions about these things, our website is a great resource to kind of start there. And then if you want to reach out to one of us to ask any further questions. Sarah, I've got one for you. I can't remember when exactly this was asked, but the question was, what kind of water source and how large would that have been?
Great question. I would say that it varied. Some of them, most of them were close to creeks, streams, rivers. I think maybe one or two were close to sort of marshy looking areas. And it definitely varied by observation, but great question.
Right. Was time of year of monarch presence matched up to surveys of floral diversity and milkweed presence, since their arrival is so much earlier in Texas than, say, Iowa?
I can say for the broad IMMP data analysis, it didn't actually overlay the monarch use data. So we have that whole range of surveys that kind of started in February, and goes almost through the end of the year. And I would welcome anyone to actually overlay the monarch use data on it, because that was just a layer we didn't have the capacity to add to this particular paper.
On a year-to-year basis, is everyone looking at weather factors during migration? This may not be completely related, but maybe that
like,
one.
Yeah, I might need to run.
Yeah. Yep, absolutely. We can come back to that one. Let's see. Did you look at the phenology split out by region? It seems it would be different in each region. I'm guessing that's, ecoregions are pretty, how the ecoregions are split up, kind of look at that, right? It's already kind of defined how they
I would say in that table that we mentioned, we kind of averaged resources, milkweed density and floral species richness and all those factors, average them by region. But we definitely could look at phenology within each region and kind of think about, peak blooms or peak milkweed presence and things like that, though we didn't have capacity to explore that either in that paper, but it's certainly a cool avenue to explore.
There's always more to explore, more questions that form. Sarah, this is another one related to the water source question. Did the less than 140 meter distance where monarchs observed from a water source apply to ponds too, or just riparian areas?
I didn't see, of the few monarchs that we observed, I didn't see any specific ponds. There were some sort of like marshy areas that they were observed with. But I'd be curious to see what that looks like in the new data set in 2023 to 2025. We've seen more monarchs, and so I think we could make stronger conclusions based on that.
I do apologize. I am manning the Q&A here, and I usually have an extra person. So let's see. Another related to this water question, did you find certain areas in the West to be near ponds? And that one's not quite as clear.
I did not.
Okay.
I only looked at monarch observations with respect to water, and we didn't quite explore milkweed occurrences, but that would also be interesting for another time.
All right. Well, here's a couple of roadside habitat questions. One was asking, does promoting habitat next to roads lead to greater deaths from autos or, I guess, auto accidents? I'll start with that one. Have you seen anything related to that?
Yeah, I think we should also follow up with our great resource about roadside habitat. We have actually one of the authors on this paper, Laura Lukens, did a great literature review on that, and kind of in sum, it's a net gain. And there's an FAQ that explores a lot about mortality and habitat quality, and production in roadsides. And while there is risk, there's more opportunity to gain from that habitat based on the research we're aware of.
I will be sure that I attach that in the follow-up email so that
Thanks,
people
Jen.
can read that. Yep. I'm not sure about that one. Let's see. There's so many great questions. Some of them are quite meaty here. This one might be a little specific, but we'll kind of see if we can find an answer to this. Because Asclepias syriaca does not occur in Texas and southern Oklahoma, was there any look at just that area as Asperula is so abundant in central and other parts of Texas when monarchs are passing through that area, with A. viridis most common along the roadsides?
Great question. I mean, the short answer is no. We only split it up by the south, west, and north. I love the opportunity to do that, and I hope that the data set is explored in that way, because those are very vast regions, and you're absolutely right. They're not, Texas is not the same as Florida, is not the same as South Carolina. So any more regional exploration is welcome, and I think there will be additional nuances when the data is explored that way.
Great. What were the non-native plants found with highest activity in the South?
Like referring to the floral resources, I would have to pull that slide
I would
back up.
assume if you've got it, otherwise we can definitely put it in the email.
Yeah, what we found most frequently for non-native species, what, in the South?
Yes.
What's the question?
Mm-hmm.
Black medic, scarlet pimpernel, and annual ragweed were the top 3 most frequently observed in bloom. And in the paper itself, which we can also send out, I think 10 or 12 are listed.
Thank you.
And I'll just point out that these are just the floral resources that were observed by the observer, and they don't necessarily indicate that the monarch was using them. But that's, I think, a really interesting interaction as well.
Good question about, you talked about the potential of rights of way, and just wondering about the process in order to get more habitat in rights of way. What does that look like?
That's a big question.
Mm-hmm.
It might look different in different areas. And so sometimes it's a ground up approach where at the township level, there's a lot of townships or counties that are contracted by the state to do the mowing, essentially, or the roadside management. And so those interactions might lead you to the right people to inform of, like, what does a milkweed patch look like? Or how can we adjust our windows of mowing so that it doesn't overlap the most critical breeding period of monarchs. And then MJV has a good roadside management handout we can also share. That's a nice 1-page resource to get in front of either habitat managers or the people who make those decisions. I think that's probably a good place to start, is finding out locally who's the best person to inform. And then our resource has some good information about mowing windows.
There's a question, is there a way to submit your land for study for the migration of monarchs? And maybe that isn't the related to IMMP, but is there a way to get your land involved in IMMP? Like if someone was interested in doing that.
Absolutely. The best way to get involved is to get yourself involved. We have free online training, and then MJV staff can be available to consult if you have questions. But this is a protocol that community scientists can apply on their land. I would start small and work your way up to the full suite of all 4 activities per se. And if that's not an option to you, to spend, a couple hours at a time or 1 or 2 hours at a time doing a survey, we may be able to connect you with people or partners, or maybe our own projects that are looking to do some surveys in specific areas, but that's much more limited.
Thank you. I will also post the link in the follow-up to the Monarch Nectar Hub, which is where the free IMMP course is located. So if people are interested in taking that, that is definitely an option. We've got one. Do any studies ever involve Senantium laevae in IMMP or other community science that you know of?
Yes, there are some research topics out there that I think there was a study. I don't want to say incorrectly, but I think there was a recent study from last year that's in our annual research review that talks a little bit more about that potential. And it was something observed in our data set, but it was lumped in with all of the milkweeds. So I couldn't tell you right now where it was located, and it was not treated differently in our data set.
Okay. Great. Well, we are coming to the end of our session. I want to post for everyone the survey. If you would be so kind as to take the survey and give us some feedback. But we really want to thank Sarah and Jennifer for presenting today. Thank you all so much for participating in today's webinar. And we hope to see you for our next webinar, which is titled Monarch Research and Restoration Highlights, Collaborations in the West. This will be happening on July 23rd at, again, 1 o'clock Central Time with Dr. Hilary Sardinias from California Department of Fish and Wildlife. Thank you, everyone, again, so much for being here. And look for the email with the answers to the rest of the questions, as well as the link to this recorded session. So thank you, everybody, and have a great rest of your day.
Thanks, everyone.
Thank you so much.